Hello, this is Tiffany Tulo, week 5. This is biochemical identification through carbohydrate fermentation lab. I'm going to be going over part 1 of my lab for the inoculation of the bacteria. Let's go over wash my hands, I have my gloves on. As you can see, I have all my supplies out, my phenol red, my specimens, and then I will get started with the inoculation portion of the So first part, I'm going to be adding the glucose powder to the tube labeled glucose. I'm going to distribute those amongst the two. Flame always being sterile, flaming the lid. And I'm going to transfer half of the powder into the tube. Careful to only do half. And I'll continue to do that with the next couple tubes until I finish here. And then I'll flame it one more time before putting the cap back on. So I have emptied out all the contents of the vials for the lab. Now I'm going to take the sterilized Durham tubes that were dipped in alcohol, now dried, and place it into the vial here, upside down into the tube. As you can see, I'm going to make sure there's no air bubbles in there, and I'll repeat that for all the other vials. Now I'm going to be transferring the broth. I'm going to do Estervassier into the phenol red tubes. So I'm going to flame this first. This is my sterilized pipette. And then I will sterilize phenol red tube. All the Durham tubes have had the air removed. And now I'm going to take 4 drops of the Estervassier and transfer them into phenol red. 2, 3, 4. And I will flame this for returning it, and I'm going to continue doing that for the remainder of the 3 tubes for fructose and maltose. Now that all my tubes are inoculated, I place them back in the bubble wrap so that I can place them in my incubation area, and I'm gonna let them stay in there for about 12 hours, and then I'll come back and check them and check for the fermentation process. It's been 12 hours since my incubation, and I'm gonna take a look at my results now. Here's the results for the glucose for each one. Here are the results for the fructose. Let's see the different processes of fermentation, and then here is the one for the mannitol. I'm going to take pictures individually and then record my results in my lab report and get it sent out.